ready? Hello, let's get it. So we are going to talk about the reproductive system and the development histology. So you have this PowerPoint on Blackboard still, and then this video to accompany, all right? So when we talk about the histology, you have your list there that you have at your desk. And I've laminated it to show the different structures that you are to know. So someone sees that? Yes, so pull them up. You have the full structure list on Blackboard, but this is just for the reproductive system, so please follow along. When we look at the ovary, okay, the thing I want to draw to your attention about the ovary is one, when we look at the reproductive system, okay, first of all, here's your ovaries. And we remember that the ovaries are not attached to the fallopian tubes. There is a ligament that spins them in. It's called the broad ligament to the lateral sides of the pelvic cavity. And that egg, under the influence of hormones, is going to rupture from that ovary once a month, right? And that is going to be swept by the fimbria of the fallopian tubes, moving the fluid in the pelvic cavity to bring it into the fallopian tube. And the sperm is going to have to travel from the vagina through the lower part of the uterus, the cervix, up into the outer third of the fallopian tube to meet that egg that ruptured and fertilize it. And then that now fertilized egg is going to come down and implant into the inner lining of the uterus called the endometrium. And so the uterus has layers to the wall. It has the endometrium, the myometrium, and the perimetrium, which is all around. The lowest part of the uterus is the cervix, and the opening is the cervical os. And so I want you to appreciate the vagina is not part of the uterus. It's a separate structure. And so the uterus sits down into the vagina. And when a woman gets a hysterectomy, she has to have her uterus removed. They cuff off the vagina, okay? And so that is the anatomy. But the fallopian tubes are continuous with the inside of the uterus, and that is how the sperm are going to travel to meet the egg and then come implant. Now, when we go through puberty, we start to have this cycle of eggs coming out, hoping that we're going to get pregnant, right? So the thing of it is that, the reproductive system is just one of the systems that we can live without. We can live and not have these things. But under the influence of hormones, for human beings, we go through a period where hormone secretion is going to become higher, and we're going to go through what is called puberty, okay? And so there is something happening here in the eggs. Ever since we were in our mother's womb, we had all the eggs that we would ever have. We have like 1 trillion of them. And through time, we have a reduction division of the chromosome numbers. By the time we go through our period of having ovulation, we probably only have about 400 eggs that actually are going to rupture to become that we hopefully would get fertilized, okay? So there's a lot of atresia. So for the female, they have two things going on. We have the ovarian cycle and we have the uterine cycle. Because as this egg, and we never know which ovary the egg is going to be rupturing from that month. It could be either ovary. But we have this development of the ovary, the egg to become dominant, and then we have the preparation of the endometrium, hoping you get pregnant. So this endometrium is going to become thick and rich to have so that the embryo can implant in it, okay? And so this endometrium has 2 layers to it. If you don't get pregnant, the top layer, which is the functioning layer, is going to slough off through the vagina, and that's your menses. That is actually in puberty, the beginning of puberty. That is the beginning of the separate cycle called the uterine cycle. So you have the ovarian cycle, which is beginning in the womb, from the time you are in your mother's womb. And then when you go through puberty, you start in puberty, you have your first menses. Now, sometimes a woman, the first egg that comes out, she does get pregnant. But usually, you don't get pregnant the very first time you have ovulation. So that membrane, that endometrium is going to detach and come off, and that show of your menses is your first beginning of your uterine cycle. So you have an ovarian cycle and a uterine cycle. And so the layer Below that endometrial functional layer is the basal layer, and that will proliferate to replenish that so that you can start the whole merry-go-round all over again for the next month, hoping maybe you'll get pregnant that next month. So that endometrium, the functional layer, is going to, the arteries are going to get longer, and the vessels that are, the glands are going to start making secretions. So now you have a thick, rich endometrium, hoping that this next egg is going to get fertilized and it'll be ready to be able to support it, and it can grow. And that beginnings then would be a part of the placenta, because the baby is not going to be breathing with their lungs, right? Those transfer of nutrients and waste from the baby to the mother is going to happen across the placenta. So there's no mixing of blood. It's going to be happening across capillaries from the baby and tissue where blood is pooling on the maternal side, and that is the placenta. And so when we look at the ovary as it's developing, this egg, okay, there are supportive cells that will be around it that will be secreting estrogen, and they will be creating solution that will support the developing oocyte. And so you will see as it continues to develop, you will get layers around of cells that will be around it, the granulosa and thecal cells. Ultimately, you will form a stalk, which is the cumulus amorphous. You will have in the antrum, in the opening area here, this little dot is the egg. You have granulosa cells around it and thecal cells that are going to be secreting the estrogen. And so all of this, and you have varying levels of development. It's not like there's one egg and everybody else is sitting back, right? There's progressively different levels of primordial follicles, primary follicles, and one is going to be the one that's the dominant follicle. It takes almost a year for an egg to become a dominant follicle, okay? And so I don't have one opinion one way or the other. I allow people to have their own opinions, but sometimes when we say that, when is the beginning of life, right? Well, you can kind of understand people's thoughts, because ever since, right, we're in our mother's womb, those eggs have been there, right? And for that egg to be rupturing from the ovary, it took a year for it to become the dominant follicle. So you were 9 months in there, a year to become the one that become the dominant follicle, right? And then the sperm has to fertilize you. You have to come all the way down, everywhere you was. He has to come from the testicles, the ductus deferens, through the shaft of the penis, vagina, cervix, uterus, all the way out to and then both of them fertilize have to come and implant into the endometrium. A lot of stuff could have went wrong, right? So nobody's a mistake, and who's to say technically what is the beginning of life, right? So I think it's an individual decision for people to be able to say that personally. But I like to share that to kind of put us to think about it, right? We don't judge people when they think life begins, right? All right, so, and I don't have to go on about where I'm going with that, okay? Continuing. This stage now is now a dominant follicle. It's a tertiary follicle. And so under the influence of hormones that are coming from the hypothalamus, we will have now this egg to rupture from the ovary, and we will have what we call ovulation. And so the hormones that are coming from the hypothalamus, we have the hormones coming from the hypothalamus, the gonadotropin releasing hormone. And that gonadotropin, stay up here with me, that gonadotropin releasing hormone is going to stimulate the anterior pituitary to release luteinizing hormone and follicle stimulating hormone. I want you to see that as this egg is developing, the levels of estrogen are going to be increasing at the same rate, okay? And when the levels of estrogen are high enough in the blood, the hypothalamus will perceive that, send out a pulse of gonadotropin releasing hormone going to the anterior pituitary, and then we will have an increase of the luteinizing hormone peaking. When the luteinizing hormone peaks, also the follicle stimulating hormone will, but the luteinizing hormone will peak higher, and that is what we correlate with ovulation. Ovulation is when the egg ruptures from the ovary, okay? So when the egg ruptures from the ovary, then this tissue that was supporting the egg will continue to produce hormones. I want you to also understand that the beginning of the uterine cycle is menses. So after that functional layer, we did not get pregnant, the estrogen levels are down, the egg did not get fertilized, progesterone levels are down. That is going to slough away from the functional layer, from the basal layer, and flush off through our vagina. But now we have to proliferate that, going for the next level. And so these cells here of the endometrium are going to continue to grow. The epithelium is going to be replenished, and the arteries are going to elongate a little bit of glands developing as we are developing a new egg. Okay? So when we get to the point where the estrogen levels are high enough, gonadotropin is going to stimulate the release of these two. This one's going to peak, and that's what's causing ovulation. Stay with me. The cells that were there are going to continue to produce something. These cells are going to now produce this, which is progesterone. And progesterone's role is to maintain the endometrium, to keep it rich and thick, hoping that this egg is going to get fertilized. So that structure, when the egg comes out, is called the corpus luteum. And the corpus luteum produces progesterone. And so that is when we now went from replenishing the basal layer to replenishing the functional layer that got off. That's the proliferative phase. When we have the shedding, that's the menstrual phase of the uterus. This is the uterine cycle. So you have two things, the ovarian cycle and the uterine cycle. So after we proliferate, now I've ovulated, and now I got more progesterone to really elongate these spiral arteries. I will start to see a lot of secretions in the glands. I'm thick and rich and ready. And what's maintaining it is this hormone progesterone. And this is what's in your birth control pills. Because as long as progesterone is high, estrogen levels are going to be low, and you're not going to have a new egg to come out. So it fakes your body out to think you're already pregnant. Okay? So that's where you have a lot of progesterone in those birth control implants or pills. And so when the egg, if it did not get fertilized, then the estrogen levels are going to continue to go down. Progesterone levels are going to go down. This functional layer is going to detach, and you're going to have menses again. And then we're going to start the whole merry-go-round again. I'm going to proliferate that layer. It's going to rupture. I'm going to have now at the same time when the egg comes out, I'm in the secretory phase where I have all these secretions. I'm ready for it to come. So you got menstrual, which is the beginning of the uterine cycle, is your menstrual period. And then I proliferate that layer, and now I secrete lots of glandular secretion to make it thick and rich, hoping that this egg is going to get fertilized. Are you good? Okay? And so when we don't have the fertilization, then that area is going to become the corpus albicans. It's going to regress and be just a little piece of scar tissue. All right? So when we look back at the ovary, now histology, okay? So when we look at a for real ovary under the microscope, this secondary follicle, if you look at your sheet, the things that you have to know from there is you have to know the Graafian follicle, okay? So that is this whole thing is the Graafian follicle. This bracket is showing the whole thing. And then we have to know the antrum, and so that's this area here, okay, the antrum. And then we have to know the cumulus oophorus, and that's this stalk here that's supporting the egg, all right? And then we have to know the zona pellucida, and so there is a clear area around the egg. Now ultimately the sperm has enzymes that is going to have to penetrate that in order to deposit its DNA into the egg, and we're going to have fertilization occur. And supportive cells around that are the corona radiata. And so when you look here, it's these granulosal cells that are creating like a rays, a crown of rays around. So the crown of rays is peripheral to the zona pellucida, and then you have the egg. All right? And there's a nucleus and nucleolus of the egg, of the ovum. So the egg, the zona pellucida, the corona radiata, the antrum, the cumulus orifice. So you can see it here. This entire thing is the dominant follicle. And when the luteinizing hormone peaks, that egg is going to rupture from the ovary. Are we clear? Okay. So those are the things you have to see there. Now, when we look at the uterus, when we're looking histologically, okay, you can see, these aren't not as easy to see, but you can see it under the microscope. But what you're looking for, again, is that the uterus, okay, we're looking at tissue here. This is the myometrium. That's muscle. You're going to be able to tell smooth muscle. And you're looking for the white. That's the top. That would be the top. We cut this. And you're looking at a slide. So the white top, if you see muscle down here, then that's the endometrium. So you're looking for that layer. If stuff is coming off of it and you see blood cells, that's the menstrual phase. Okay? So when you look here, you can see there's detachment of the epithelia. You see blood. This is the menstrual phase. Okay, this is menstrual phase. Are you good? Mm-hmm. And that's usually, we got it. Okay, menstrual phase. So that's what you're looking for. This is muscle. Okay, and I know you can't see it as good, but if you were looking, it would look darker, and you could see this is smooth muscle. All right, and you can appreciate this is epithelia. So you're looking for the surface, and that's what's going to tell you what it is. And then the next phase, we slough it off. We have to proliferate, right? So we have the proliferative phase. So now you see it's flat. We're making a new layer of the endometrium, the functional layer. You can see the muscle different from where you see glands and arteries. They're straight now. They're just straight. We don't see secretions in them. This is flat. This is proliferative, okay? And then when we look here at the secretory phase, now we see this is the endometrium. Okay, this is muscle, the myometrium, and then we see there's secretions in the glands. And you're going to be looking at your slides, so you're going to be able to know. It's multiple choice. So you're going to see something that's the only thing that. And the other things is not going to, this is not a salivary gland. This is not, concretions with those crystals in the prostate. It's not the liver. Like, if you practice your stuff and go through your flashcards, we know how to do it. You're going to know it. You're going to know the stuff you're supposed to see, and that's what it's going to be, and the answers are going to be there, okay? You can't touch the microscopes, all right, at all. So when you use the scopes for the test, it's probably best to just do one eye, because you can't move it, you know what I mean? We used to have a thing where we covered one of the oculars, but I don't know where those things are. And they said, we don't have those. I said, they had them. I'm going to find them. Because it just helps you to just not, you know what I mean?
Not one to touch it.
Well, yeah, So are we clear on these phases?
Yes.
Mm-hmm. All right. So then when we go to the breast. So the next thing we have here is the breast after the uterus. And with the mammary gland, we're talking about here, we have these glands. These are modified sweat glands, but they're exocrine because the secretions of the milk that they're being made here under the influence of prolactin, that hormone coming from the anterior pituitary, is going to cause the development of these, now we call these cells that make the milk alveoli. Okay? Excuse me. Oh, excuse me, tape. Sorry. Okay. I'm breaking my rule because can you hear my airway closing?
Mm-hmm.
No. What? So silly. Okay. Bring it in. Okay, let's look at it. So you're going to have to be able to tell a breast that is active and a breast that's lactating. All right, so here's a line showing you. So we saw, those of us who went to cadavers, that the breast has a lot of adipose tissue. Stay up here with me. Above this line is a non-lactating breast. All right? And so the ducts that we have under the influence of when we're pregnant, and now we're going to have this increased production of prolactin. Then these ducts, where we don't see in the non-lactating, we just see the ducts, we will have now the development of these alveoli. So these little structures, we had a cini in the salivary gland, but now we call these structures that make the milk alveoli. and they will come into ducts we call the lactiferous ducts that will end upon the nipple and the areola, the pigmented part of the breast. And so when we're not lactating, you don't have this expression of these alveoli. So it just looks like these little connective tissue. But you see fat, but you don't see these. Okay? And if you see, it's very little and you don't see anything inside of them. So that is a non-lactating breast. When you look at a lactating breast, you're going to see these alveoli and you'll see secretions in the lumen. And that's a lactating breast. There will be fat there. There's septations. Okay, there's suspensory ligaments that are holding the breast, anchoring it to the pectoral muscles. All right? That's why you got to support your stuff, because like my cousin says, hold on, I got to tuck my breast into my belt. She's all blessed when we was little, but now it's like, woo! So you got to support them, okay? Because that's all you got holding up, and gravity is a mess, okay? And it'll be going down south, baby. Okay? Are we good? Yes. I want you to appreciate that around the alveoli are these myoepithelial cells. These are muscles that are going to contract and squeeze the milk out. So oxytocin is going to stimulate those cells, those muscle cells. So we say oxytocin helps for the forceful contraction of the uterus, and it also works on the muscles to help the milk let down. So prolactin is to make the milk, and oxytocin is for stimulating these muscles around the alveoli to let the milk down. Is it good? Mm-hmm. So when we look histologically, we can see this is an inactive breast, okay? And I know when you look at it under there, it's going to be good. And when you look at it, it's sort of a little bit better. But get it, okay? This is fat out here. There's septa between there. This is not a salivary gland with all those acini and ducts. This is just an inactive breast. It's multiple choice. So you're going to say, oh, yeah, okay, this is what this is. Are you getting it? Mm-hmm. Mm-hmm. It's going to be different. So it's not going to be like, oh, I can't tell. Look at the choices, okay? And it ain't going to be Mickey Mouse. And that's 65% of it. So I mean, if you did not get an A on a lab test, I need you to come into this knowing you can get it. But you're not going to get it by nothing. You got to practice. I got flashcards out there, and just practice them. You can do the multiple choice ones, but you just got straight flashcards too, guys, okay? Now, when you look at a breast that is active, okay, I see the septa, I see there's fat, but now I see something in the lumen of these alveoli. This is a lactating breast. Got it? Mm-hmm. Uh-oh, not that. Okay, so now we're moving on down the line, and we're going to go to the testes. So we talked about the testes. Testes here. We said this is where spermatogenesis is occurred. And so we said these are the seminiferous tubules, right? If I cut one of these tubules, then that's what it's going to look like, right? I said that from the tubules, the sperm are going to go to the next structure, which is the epididymis. Do you see it? We saw it on the models. I'm doing 1, doing 2, doing 3. Okay? I hope you get it. This is how it works. This is how your brain works. So that's why I always ask you, and I know I'm on tape, but I always ask you that everything I'm doing is a part of layering the pizza. Okay? So pay attention. If you only got the last piece of the pizza, no wonder. I just like my 3rd time saying it, man. Okay? I'm repeat and repeat here. I'm sure y'all probably like, okay, move on. No, I'm going to say it 4 more times. Okay. Leave it alone. Epididymis. This is where the sperm are going to mature and learn how to use their tail. Okay? And so they don't know how to swim here yet, but they have the flagella. So they learn, they mature, and they get the function up there. And see how much convolution there is. They spend a lot of time here. And if a man is producing sperm and he does not have emission, right? He does not have an orgasm, he does not have an ejaculate, then those sperm that are produced get resorbed by macrophages here, okay? And so that is where that happens for storage and maturation of the spermatozoa. So now when I look here in a seminiferous tubule, which is these, where sperm are being produced, it gives ovary. Because in the periphery, you got the spermatogonia. And they're progressively developing until they become, here, we're zooming, this is the seminiferous tubule. So they're progressively undergoing the reduction of the chromosome number, and then they become the spermatids. So the spermatozoa get more development, and then they become spermatids. And then they travel on to the epididymis, okay? And you can see the tails here in the lumen. All right, so between the interstitial cells, we have these cells that are in the Leydig cells. They are between the tubes, and these are the ones that are producing testosterone. So in the male, the luteinizing hormone is going to stimulate them to make testosterone. Luteinizing hormone in the female stimulates the egg to rupture from the ovary. And the follicle stimulating hormone in the female is stimulating these cells here that I told you make estrogen. The follicle stimulating hormone is going to stimulate these cells here, the thecal cells and the granulosa cells that are surrounding and supporting the egg. Those are going to be what the follicle stimulating hormone is going to cause to release estrogen to help this continue to develop, okay? These cells, all right? All of these cells, the thecal cells and the granulosa cells. Those are what the follicle stimulating hormone stimulates in the female. For the male, the follicle stimulating hormone is going to stimulate the developing spermatogonia, okay? And so that is going to also, the follicle stimulating hormone, the way it does that is that there are other cells between these, which are the nurse cells, which are the Sertoli cells. And I told you those Sertoli cells are the ones that are going to be the ones that secrete a protein that keeps that testosterone in the area of the developing sperm. They are the nurse cells. And they can't allow blood to come into here because there is a blood-testes barrier, because these guys were not involved in the maturing of the T cells and the B cells, so our body recognizes these as foreign. And so the spermatogonia, as they're developing, they must be protected from antibodies and white blood cells, so there is a blood-testes barrier that these Sertoli cells keep nutrients in the presence. They make a protein that binds testosterone, an androgen binding protein. And so again, these are the nurse cells or Sertoli cells. And they make something called an androgen binding protein, which keeps testosterone in this area for the developing sperm. The androgen binding protein. Androgen binding protein. And that is what FSH stimulates for the male. And LH from the anterior pituitary stimulates them. And the Leydig cells make testosterone. Yes, we're going. We're going. It's a little physiology, okay? So we do. Are we good? All right. So when we look at it histologically again, we saw, okay, this is the seminiferous tubules. We're not going to think this is a salivary gland. We see the sperm in there. Come on. You know this is the male, right? And we're going to be looking at them. It's not going to be our first time. So when we come to that slide, we're going to say, okay, what do they want me to know? Let me read the question. And then you're going to look at the answers and you're going to bubble it. So on the test, you're going to have a sheet to write your answers for the cats and the kidneys. And then you're going to have a Scantron, right, to bubble the answer for the microscopes. So you'll have two things on your clipboard, okay? All right. So if there's seminiferous tubule, if it's there, spermatozoa, interstitial cells. All of these are between the tubules. All of these are examples of interstitial cells of Leydig. Those are the ones that make the testosterone. The L in LH and the L in Leydig. And those are the ones that make testosterone. Is it good? Come on. Then we go on to the next one, which is the sperm. The spermatozoa, all we got to know is the head and the tail. Tail is the flagellum. Is it good? Okay, then we go to the prostate gland. Again, with the prostate gland, these concretions are calcifications you see in older men. And they have these concentric lamellated bodies, look like little crystals. You're not going to get that from anything. You got to know that's the prostate gland. Is it good? And then here they're making secretions. This is making the second largest amount of this liquid part of the ejaculate, which is called what? Starts with an S. Semen. And what was the other gland that gives the semen? The seminal vesicle. The seminal vesicle. The seminiferous tubules are these. The seminal vesicle, okay, is this, right? So the prostate gland gives part of the semen, and then these seminal vesicles. Remember we saw them behind the bladder? Mm-hmm. Those teardrop-looking things, remember? Mm-hmm. Yes? Yes. All right, see? It's layering the pizza. We already synced him. And then we come to the penis. Okay? So we remember that here's the bladder, there's the prostate gland. There's a little membrane that is going to support all your pelvic contents. As the tube that drains the bladder comes through the shaft of the penis, right? Then it is surrounded by this spongy tissue that can become filled with blood so that the penis can become erect. And so that is directly around the penis is called the corpus spongiosum. Okay, the corpus spongiosum is the one directly around the urethra. So there's the urethra. And then laterally, you have paired structures here and here, the corpora cavernosa. And those are here, all right? And there is a median septum between them, okay? When you look histologically, the epithelia of the urethra is transitional, okay? And so that is, again, the same thing in the bladder. It is a modified cuboidal epithelia that can flatten out when the bladder gets filled with urine, or it can go back like this. So it helps to accommodate. It can transition. And then we have around the urethra the corpus spongiosum. And then we have on both sides here the corpora cavernosa and a median septum. Is it good? Mm-hmm. So now we come to our last topic, which is development. And for a couple of these, I want to show a little bit of slides, okay? We're doing good. And so we have here, we have the cell. When it gets fertilized by an egg, as I'm showing, you can not appreciate when you have the egg and the sperm come together and fuse. The cytoplasm is going to be mostly coming from the egg, but they will share it, and you will have a single cell membrane, and leaving only, first you'll have two separate nuclei in the center, and once they have fused to form a single nucleus, now the newly developed organism is called a zygote. And so this is what we're seeing. And progressively, these cells are going to differentiate and multiply to ultimately form our three primary germ layers. And so you will have these divisions. And so you will see the 2-cell stage, the 4, 8-cell stage, 64, we call it the morula or the blastula. And the inside of that, as we have this invagination, we're having this as the blastocoel. And so we will have this early gastrulation. You will have this invagination, which is going to also leave this structure here called the blastopore. And this structure here is going to have a migration of some of these cells coming from here. This area is going to be now what is going to be the formal endoderm. And the migration of cells from this invagination will come between here, and that will become the mesoderm. and the outer area then will become the formal ectoderm. And from each of these germ layers, you have specific tissues that are us, right? Ectoderm, your skin. Endoderm is going to be like your organs. Mesoderm is like your muscles. And there's various structures that come primarily from those different germ layers to make us. And so, as I'm saying, the anticentron here is going to eventually become the cavity of the digestive tract, and the blastopore we saw here is going to be the anus. And so when we look here on this developing starfish, that is what we have as a slide. You have on your sheet, you have the unfertilized egg here that we can see. We have the 2-cell stage. We have the 4-cell stage. We have the blastula morula, which is the 64 here. This should be over a little bit, all right, or here. And then we have the endoderm, ectoderm. We don't have a representation here. These are the things that are on there. I'm going to cut this a little bit because these are the things that you're going to be able to see. So here, what you do not see, the obvious nucleus, this is the zygote, and those are the big ones. So we have the other big one, we have the blastopore, okay, so that is going to be the gastrula, and the other big one is the blastocele. We don't have it labeled, but you're going to be we could come in this area. So I'm going to get you some little bit better. I don't know if I have that, but we're going to make sure I got everything that's on here in a representation on a different slide. Is it good? Mm-hmm. Finally, let's go back. You're doing good, class. I just want to show a little video on that, and I want to say something about then when the baby is developing. So we talked about the endometrium. And so there is a maternal side and there is a fetal side. And so for the maternal side, the maternal portion, you're going to have those arteries, but the blood is not going to mix. It's going to pool into some areas. There is going to be the formation of tissue that these capillaries from the fetus are going to come into, and the tissue is going to be around it, supporting it. And so things are going to come from the fetus into this area with diffusion, and then the transfer of the waste are going to be going back to the mother so she can utilize her systems to get rid of things, and she will be bringing oxygen. So when we look at the umbilical cord coming from the baby, away, right? The umbilical arteries are coming from the baby. You see they have the deoxygenated blood, and they're coming up, and then they will be getting the oxygen from the blood coming from the mother. And waste and nutrients will be transferred in that way. And so then what is coming back to the baby from the mother is going to be the umbilical vein. And so you see that that is red. So you have two in the umbilicus and the umbilical cord. You have the two umbilical arteries and the umbilical vein, which is going to have the oxygenated blood. And so when we look under the microscope, now you can see that structure that is around and supporting the capillaries from the baby is the chorionic villus. So this tan epithelium. And then when you see the blood vessels around that, inside of there, then you know that is the fetal part. Those are the chorionic. And then the other part, the spaces where you see blood, that is the maternal side. Okay? All right. And so again, the tissue is the chorionic villus, and where you see the blood, that is the blood vessel. That is chorionic villus too, around it. But in these spaces, that is going to be where the maternal blood is going to be. Okay? All right. All right. So now, when we look at this umbilical cord, we see now the two umbilical veins and the umbilical artery. And so all of this supporting tissue, this gelatinous connective tissue that's supporting the umbilicus, has a lot of stem cells. So we say we want to get the cord blood because you have a lot of mesenchymal cells that have not been differentiated. And so they could use those in tissue culture or in cancer to give different stimulating factors to those stem cells. So we want to use those for research, and we use those. They can make a lot of different cells, your nervous tissue, your white blood cells, and so we can use them to study them in the lab to see and differentiate them into different types of things. They use cord blood and differentiate it for the joints, for your bones. They inject those things. So stem cell utilization, therapeutics, and research is important, and because they're new and they're fresh and they haven't been committed. So they are still not a nerve cell or epithelial cell or a muscle cell. At this point, you can give some stimulating factors and grow them up in tissue culture as a nervous cell or a bone cell and use that in somebody that has an injury or needs to have some growth, okay? So that tissue, that supportive structure that is around is called Wharton's jelly, all right? So when we see the slide, we see the two umbilical arteries and we see the umbilical vein, all right? And this is the oxygenated and this is going to be the deoxygenated, and that is Wharton's jelly all around it. Is it good? All right, so that's what you have to know. So let's go back. I know we're running out of time. I have 20 minutes? Yes. Okay, this here, again, we're not going to show all of it, but I just want to let us get a little bit of a visual of this. So let's just see real quick, and I'm going to speed it up, what it looks like for development.
The bilamellar embryonic.
Real quick.
By the end of the second week of development, the bilamellar embryonic disc, consisting of the hypoblast and epiblast, has formed. Throughout the third week of development, this bilamellar disc differentiates to establish 3 primary germ layers, in a process known as gastrulation. Around 15 days after fertilization, a thickened structure forms along the midline of the epiblast, near the caudal end of the bilamellar embryonic disc. This is called the primitive streak. At this stage, the formation of the primitive streak defines the major body axes of the embryo, including the cranial end towards the head and caudal ends towards the tail, as well as the left and right sides of the embryo. At the cranial end of the embryonic disc, the primitive streak expands to create a primitive node which contains a circular depression, known as a primitive pit. This depression continues along the midline of the epiblast towards the caudal end of the streak, forming a primitive groove. Once formed, cells of the epiblast migrate inwards towards the streak, detach from the epiblast, and slip beneath it into the interior of the embryo. This process is known as invagination. The first cells to invaginate through the primitive groove invade the hypoblast and displace its cells. The hypoblast cells are eventually completely replaced by a new proximal cell layer, which is referred to as the definitive endoderm. By day 16, the majority of the hypoblast has been replaced. The remaining cells of the epiblast are now referred to as the ectoderm, and forms the most exterior distal layer. Some of the invaginated epiblast cells remain in the space between the ectoderm and newly formed definitive endoderm. These cells form the germ layer, known as the mesoderm. Once the formations of the definitive endoderm and mesoderm are complete, epiblast cells no longer migrate towards the primitive streak. Throughout gastrulation, the ectoderm continues to form from the cranial to the caudal end of the embryo, establishing 3 distinct primary germ layers throughout the whole embryonic disc. The gastrulation process is finally complete.
All right, so that's gastrulation. Now, this is, real quick, let me do one more. This here is how the placenta develops, okay? Almost. Real quick. Thank you.
Development of the placenta begins during implantation. When the blastocyst adheres to the endometrial epithelium, trophoblast cells differentiate into an inner cytotrophoblast layer and an outer syncytiotrophoblastic layer. As the cytotrophoblast proliferates, newly formed cells migrate into the syncytiotrophoblastic and lift their cell membranes, forming a rapidly growing multinucleated mass. The cytotrophoblast secretes proteolytic enzymes, and the syncytiotrophoblastic sends out finger-like projections, allowing the blastocyst to embed into the endometrium. Lacunae, or spaces, begin to form within the syncytiotrophoblastic. As the syncytiotrophoblasts erode the endometrial blood vessels and glands, the lacunae become filled with maternal blood and glandular secretions. Isolated lacunae fuse to form a network through which maternal blood flows, thus establishing early uroplacental circulation. Toward the end of the second week, small projections of the cytotrophoblast begin to expand into the syncytiotrophoblasts, forming primary chorionic villi. Early in the third week, extra-embryonic mesoderm grows into these villi, forming a core of loose connective tissue, at which point the structures are known as secondary chorionic villi. By the end of the third week, embryonic blood vessels have begun to form in the extra-embryonic mesoderm of the secondary chorionic villi, transforming them into tertiary chorionic villi. Cytotrophoblast cells from the tertiary villi grow toward the deciduum basalis and spread across it to form a cytotrophoblastic shell. Villi that are connected to the deciduum basalis through the cytotrophoblastic shell are called anchoring villi. Villi growing from the sides of the stem villi are called branch villi. These branch villi are surrounded by the intravillous space and will serve as the main sites of exchange between mother and fetus. By the fourth week, fetal blood flow is established. Poorly oxygenated blood is transported to the placenta from the fetus via paired umbilical arteries. These arteries send branches into the chorionic villi and divide capillary networks at the terminal ends. Carbon dioxide and waste products are removed from the fetal circulation across the placental membrane to the maternal blood and the intravillous space. Oxygen and nutrients are transported across the placental membrane from the maternal blood to the fetal capillaries. Oxygenated fetal blood travels back through veins, converging to form a single umbilical vein that goes to the fetus. Blood returns to the maternal circulation through endometrial veins, forced out by the pressure of incoming arterial blood. The placenta nourishes the fetus throughout development and is delivered shortly after parturition.
Okay, so let's see what it's like to birth the placenta. So about 30 minutes after pregnancy, now that placenta has to be detached and come out. So let's look at it.
In our previous videos, we delved into the interest wonders of fertility and fertility. Now it's time to embark on the next chapter of the journey.
Hold on.
After the baby is safely born, it's time to deliver the placenta. Usually, it delivers within a half an hour. As it comes out, hold it in both hands and twist it to bring all the membranes together. There should be minimal bleeding that stops quickly.
Sometimes though, a piece of placenta or membrane is left behind in the uterus.
This can lead to continuous bleeding and infection. This video shows how to examine the placenta for any missing pieces. Let's first see what happens to the placenta from inside the body. After the baby is born, the uterus contracts, and soon after, the placenta separates from the wall of the uterus. A clot of blood forms where it is separated. The uterus continues to contract, closing off the bleeding vessels as the placenta moves down and through the birth canal to deliver. Sometimes, though, pieces are left behind in the uterus. Even a small piece can prevent the uterus from contracting, and the vessels will continue to bleed. This can lead to hemorrhage or infection. It's important to carefully examine the placenta. The smooth and shiny side faced the baby in the uterus. The cord attaches on this side and then spreads out into many deep blue blood vessels. The maternal side of the placenta is the rough, red, meaty side that is attached to the wall of the uterus during pregnancy. To check the placenta for completeness, hold the placenta in the palms of your gloved hands with the maternal side facing upward. Look carefully to be sure that all the lobes are present and fit together. Check the edges of the placenta for torn veins that could indicate extra lobes. There should be no missing piece or sign of breakage when the placenta is stretched flat over your hands.
Inspect the membranes for completeness. They may appear ragged or some may be missing.
If a portion of the maternal surface of the placenta is missing, it's likely that the placenta pieces are still in the uterus. It can be dangerous for the mother if any parts of the placenta are left behind. She is at risk of postpartum hemorrhage and infection because the uterus will not be able to contract completely.
And so
If
in
you
that,
see anything
they're gonna have to
confusion
go
on
in
the
there
exam,
and do a debridement and make sure they get it. When I was in school, that's one thing they would let us do. Count the cotton leaves and say, "Ooh, we gotta do it." So now we're almost done, okay? All right, so now let's come over here.
So I stop.
Yes, thank you.